All right, welcome everyone. So we're going to begin a new series, a new kind of set of videos and lectures. And what we're going to be looking at in these several videos upcoming are going to be transport processes, right? So the previous set of videos, we looked at filtration processes. Okay, so if you haven't watched the filtration series, make sure you go and you view that. But what we're going to be doing here is basically looking at after we have filter the blood, right? So after we've taken the blood from the glomerulus and we filtered it into that capsular space, and in the previous series, right, we talked about some of those different pressures, hydrostatic pressures and osmotic pressures. After we've filtered that blood and we have this capsular fluid or this tubular fluid, how do we ultimately move things, back into the blood? How are things moving across cells? Things like that, right? So that's what we're going to be looking at. We're going to look at general processes. We're going to talk about specific examples as well. So specifically sodium and specifically water and several others. And then we will go from there, right? So if we look at a few mechanisms by which we can transport substances from one side of the cell to the other, so meaning from the lumen of the tubule, right? So maybe inside the proximal convoluted tubule to now inside the blood, we can go two ways. One is called paracellular or paracellular transport, and one is called transcellular. You may see the term transcellular. You may also see the term transepithelial, right? So if you see the term transepithelial, that means the same thing as transcellular, right? Just two different ways to describe the same thing, right? So paracellular, the term para means beside, right, or next to. So when substances move paracellularly, they're moving between two adjacent cells, right? So they're moving next to or beside one given cell, right? So here's one cell, here's another cell. We're moving in between those two cells, right? If you're moving transcellularly, right, this term trans is meaning like across, right? Or from one side to the other across the actual cell itself, not between, but across. So here we're moving, so maybe inside of the lumen of a tubule, proximal convoluted tubule, we're moving through that given cell, right? Not between it, not beside it, but actually through it or across it, right? So for this one, transcellular, we're going to be moving across that basal membrane, right? And with this, there will be various proteins that are embedded within the membrane of the cell that are assisting in the movement of that given molecule, right? Maybe it's sodium, maybe it's chloride, maybe it's glucose, maybe it's some other molecule, right? So we'll move these things. Again, they're moving from inside the lumen. So again, the lumen, think of as the opening in the tube, right? So the your blood is in the lumen of your blood vessels, right? The opening of the blood vessel. So the lumen of our tubules is just the space where the fluid is collecting, right? Within that lumen, there are the various cells that are lining it, and those cells are what are going to control and regulate the passage of water and sodium and glucose and amino acids and all these different things from one side of the cell to the other, right? During this reabsorption transport process, we're moving things from the lumen, so that luminal fluid, ultimately back into the blood, specifically these peritubular capillaries, right? And so they're going to move back. And then for these processes of reabsorption of sodium and water and many other things, a lot of this is occurring in the PCT, so in the proximal convoluted tubule, right? Now, the reason for it occurs throughout the length of the nephron, okay? But primarily, most of it is occurring in the PCT. The reason for this is because of microvilli, right? So recall microvilli. Microvilli are those small kind of finger-like extensions of the plasma membrane of cells. And if you recall what they do, right, they serve to increase surface area, right? And so in this case, increasing surface area for the reabsorption of these various solutes, molecules, things like that, right? These various substances. So this micrograph, this histological section you can see off to the right of the screen, this is a view of the proximal convoluted tubule. right? So remember, it's very convoluted. So these individual sections, they're likely all part of the same tube, but because it's very convoluted when it's sectioned, you get multiple different views, right? But one thing to note here, again, you can see the individual tube, you can see kind of that simple cuboidal epithelium, but you also see this kind of ruffled end on the inside, how it kind of projects into the lumen. That's the microvilli, right? If you look at a section of the distal convoluted tubule or the collecting duct or parts of the nephron loop, you likely will not see that kind of ruffled, fuzzy end kind of on the inside of this lumen because those don't have microvilli, right? Those regions don't have microvilli, right? So here you can see kind of a schematic of our tubules along with the peritubular capillaries that we were referring to, right? So here's our capillary, right? Here's our peritubular. Peritubular just means beside the tubule or around the tubule, right? So here's our peritubular capillary. Here you can kind of see the schematic of it, this red line, right? That's the peritubular capillary. And then here's our lumen, right? So this over here, this would be the glomerulus. This is the capsular space, right? So this Bowman's capsule with the space on the inside. And then here is our PCT, right? Our proximal convoluted tubule. And notice a few things, right? Notice one, the presence of the microvilli on the apical surface, right? So meaning the surface closest to the lumen, that luminal surface of the cell. Notice the microvilli, right? You can see those little small finger-like extensions, right, all along this apical border, right? And then notice also it's a simple cuboidal epithelium, as we said, right? You can see that same epithelium here. And then last thing, notice the two different transport mechanisms, right? So meaning we're moving things either paracellularly, okay? So here you can see paracellular movement, right? Between the cells, movement of substances between epithelial cells, or transcellular, again, also called transepithelial, moving across the cell. And notice here it's utilizing some type of protein, right? So it's going to be a protein channel, maybe it's a protein carrier, right? Remember, both of those are involved in the movement of some type of molecule, right? Now we'll talk about different types of movement, right? Some of them are passive processes, right? So remember, from previous lectures you've done, previous topics, um, remember passive is not requiring energy. It is down a concentration gradient, meaning from high concentration to low. Other processes that we'll look at are active, right? Remember, active is requiring energy in the form of ATP, moving something from low concentration to high concentration, right? So we'll kind of look at both of those, right? Now, one kind of important concept to understand is something called transport maximum and renal threshold. So these are different concepts, but they're related, right? So transport maximum, this is the amount of some substance, some material, okay, so something in the filtrate that can be absorbed and secreted across the renal tubule in some given amount of time, right? So let's just say the amount of some molecules, so take glucose here for as an example, the amount of glucose that can be reabsorbed from the tubular fluid back into the blood over some given amount of time. So for glucose, it's 375 milligrams per minute, right? That's the amount that can go from the tubular fluid and be taken out of the tubular fluid, tubular reabsorption, back into the blood, right? 375 milligrams per minute, right? This other term called renal threshold is similar, or it's related, but it's slightly different, right? So this is the plasma concentration. So the concentration of said substance, in this case glucose, the concentration in the plasma in which it will begin to appear in the urine, right? And so the threshold is the maximum amount that you would have or that you could have without it beginning to appear in the urine. And then once you exceed that threshold, then it could affect the amount of that substance that you find in the urine, right? So this renal threshold is going to affect the transport maximum, right? So if you have a given amount of some substance, in this case glucose, and you exceed the renal threshold, you'll begin to see that um, see that particular substance appearing in the filtrate, and because it has exceeded the threshold, the transport maximum will be exceeded, and the transport maximum is essentially dependent upon the various proteins that are going to be used to help move that substance back into the blood, right? So essentially, think if you've got too much of the substance, the molecule in the blood, then that amount is going to end up in the filtrate. And if you have too much of it in the filtrate, it's going to be more than what the transport proteins can handle. So some of it will get reabsorbed, right? Some of it will be brought back um, uh, into the blood, but not all of it. So you will be unable to reabsorb all the glucose, right? And that amount that you don't absorb, right? What do you think happens to it, right? Well, it's going to end up in the urine, right? So in the context of glucose, as our example, you end up with something called glucosuria, right? Glucosuria. Sometimes you may see the term glycosuria, right? So GLUCO, or you may see it as GLYCO, right? Glycosuria or glucosuria, right? The roots of this word here, gluco, is obviously referring to glucose, and anytime you see that ending uria, right, that means urine, right? And so in this case, glucosuria, you find glucose in the urine. Proteinuria, you find protein in the urine. Hematuria, you find blood in the urine, and so on, right? And so this is when you have extremely, extremely high plasma glucose concentrations, right? Um, and so it will exceed that renal threshold, it exceeds the transport maximum, and you end up with that glucose in the urine. This is detectable via a urinalysis, right? Now, something related to this, and you find this often in diabetes, right, in patients who are diabetic, because of this, because of the poorly regulated because of the poorly regulated blood sugar, it ends up increasing in the blood, and then that amount spills over into the urine. Now, one other thing that you'll see several things in patients with diabetes, but one thing you'll see kind of in relation to the glucosuria is increased urine formation, right? Increased urine formation, and so this is called polyuria, right? Polyuria. So poly meaning lots or many, and then urea meaning urine. So you make lots of urine, right? So the reason for this is that as you spill the glucose over into the um, urine, right, into the tubular fluid, it's exerting an osmotic, gradient. It's exerting an osmotic force and pressure on the fluid, and it's helping to pull and retain some of that fluid that could have gotten reabsorbed back into the blood. It's helping to, or it's acting to keep some of that fluid in the tubule, and then that tubular fluid is ultimately what's going to become urine, right? And so something you'll see um, with patients who have diabetes is polyuria, the excess loss of urine. And then related to that, they end up with something called polydipsia, which means excessive thirst, right? And then there's another one called polyphagia, in which they have an increased appetite and hunger, right? So those are kind of the three P's of diabetes: polyuria, polydipsia, and polyphagia, right? So let's stop here, kind of wrapping up with those kind of basic transport mechanisms and whatnot. And what we'll pick up with is we'll begin looking at nutrient reabsorption, and we'll look at some specific examples with sodium, with glucose, with proteins, and then we'll move on beyond that afterwards and begin looking at water as well.